All right, I think we can slowly get started. Everyone's settled in. Please help yourselves with lunch as I speak. Don't hesitate. Thank you all for coming here today, and thank you for the invitation, for having me here today. The purpose of today is to give you all an introduction to PET-CT imaging in preclinical setting, what it can mean for oncology research, and what are kind of the highlights on how to get started with preclinical PET-CT imaging. So I am aware that here at the ICBR you have a facility with optical imaging, and I just wanted to check with the audience, maybe by a raise of hands, who is experienced already or knows something about preclinical imaging. Have you used it in your research? So who feels experienced with preclinical imaging? So that's almost a third. Okay, that's good. It's good to know, just to know where I should get started with the material. So the purpose of today will really be on a couple of things: introduction to this preclinical imaging at large, then zooming down into what PET-CT imaging is, some of the highlights, then what PET-CT imaging can do in oncology research, and lastly, how to get started with your lab. Here we are. So an image always says more than 1,000 words. I'd like to start the presentation just by sharing some examples what you can achieve with these types of imaging modalities. PET-CT imaging or oncology is actually one of the original largest applications of PET-CT imaging. There's a wide variety of cancer preclinical animal models that can be imaged for the validation of your animal models, for the tracking the therapy that you're trying to build for these models, etc. So here on the left, from left to right hand side, we have an example of breast cancer metastases, where you can have the full vision or the full view on your metastases throughout the body of the animal. Xenograft or orthotopic models that can be imaged. Just with CT alone, there's plenty of things that you can do in terms of tumor volume measurements. Same with contrast-enhanced CT on hepatocellular carcinomas in the liver, and so on. To get things going, so first I wanted to cover a little bit more what preclinical medical imaging exactly is, and then zooming down further. So in terms of medical imaging, on the top row here we would have what is imaging typically used in a clinical setting, where you use imaging for in a single patient for your diagnosis, during the treatment, sometimes even to deliver the treatment if you're talking in the theranostic field, and in the follow-up. However, in a preclinical setting, imaging is more used in a volume setting, in the sense that you want to use multiple animals to gain statistic relevance, to track a certain known condition that you want to track. This could be the tracking of your disease model in the animal to validate your animal model, to validate a specific cell line. But this could also be to track the effects and the efficacy of your therapy that you want to achieve. The example here shown beneath is again a simple contrast-enhanced CT image, where over time the tumor volume can be measured, quantified, and the efficacy of a proposed medicine can then be proven, basically. Imaging in clinical, but as well in a preclinical setting, can be broadly split in two groups. You have the anatomical, let's say, cluster, and the functional cluster. Anatomical is really looking at, of course, the structure and the physiology of your animal, looking at the bone structure, where functional is really zooming down even into the picomolar level, showing the metabolism inside the animal, showing the different processes that can happen in the animal, the biodistribution of different compounds that you would like to image. And the different modalities that exist out there can be split in these two broad groups. You might see or recognize the optical imaging one, which is here available, or as far as I understood here in the ICBR available as well. CT is, of course, obviously an anatomical scan. Together with contrast agents, you can get into more functional areas, but it's the true power of PET and SPECT imaging that shows, or the true power of PET and SPECT imaging is really the functional aspect. And combined PET-CT imaging, so the hybrid formats, the multimodal formats, you can have the best of both worlds. The added value, as I mentioned, beyond the CT, where you first have the structure and physiology of the animal, is really with PET and SPECT, where you can go into the metabolism, the drug distribution of a novel compound, molecular pathways, also molecular targets, receptors, and binding sites, and not only visualize these processes, but really quantify them at a very accurate level. One comparison, there's a slight comparison that can be made between the optical and the PET and SPECT imaging, but where PET and SPECT truly go further is in the sense of having a much better quantification potential at any depth in your animal. Optical, there's only so much depth penetration inside, and also at a very or at a much higher resolution. So what is exactly nuclear imaging? Nuclear imaging is the combination of PET and SPECT imaging. They both are similar. As all the other imaging modalities that we just talked about, it's non-invasive, but it uses specific biomarkers. So it uses radioactive probes. You take a radioactive isotope, an element that you bind with a specific ligand. Could be, for example, fluoride 18, which is a isotope that you bind with FDG, type of glucose, which is then metabolized throughout the body. If you then inject this tracer, as it is called, inside an animal, the FDG gets metabolized, but the little isotope that's bound to this sugar will still emit gamma rays, which are then captured by the PET camera. And it's this way that you can really image the entire processes inside of animals. FDG or F18 FDG is just one of the many different configurations that you have between different isotopes and different ligands. You can hang isotopes on antibodies, on different sugars, on specific monoclonal antibodies. There's a whole range of those. So in terms of what a type of workflow looks like for PET-CT imaging, it's fairly similar to what you would have for optical imaging. First, there's the tracer injection of the animal. It's typically a 10-val injection, typically a couple hours, or depending on the study, a couple hours per day before the actual imaging. The tracer will distribute inside the animal and will metabolize. For example, with the FDG study, this is, or the 18 FDG, it's very much used in oncology research, as obviously cancerous tissue metabolizes the sugars quite heavily. The image is then acquired, and we can have a look at how that works in just a minute. And then an image is reconstructed to give you the fully tomographic 3D data, basically. So the exact way how it works is, as you have here on the left-hand side, this F18 isotope that I just mentioned before. Basically, there's a radioactive decay, and a little positron element splits off from the element, and the F18 will decay to an oxygen atom. And then this positron will annihilate with an electron. And basically, you would have two gamma rays that disperse in 180 degrees in a single line. And it's these two lines that are then detected by detectors in the PET camera. And if you have a coincidence hit on one side, a coincidence hit on the other side, you can draw this line basically, and you can precisely pin where this coincidence, as we call it, occurred. And by having billions and billions of these coincidences, you get a whole PET image at basically a very high resolution and a very high sensitivity. So even within the complete interior of your animal, you would be able to capture these coincidences. So this was just roughly broadly on PET imaging in a preclinical setting, what it exactly is. Now to move on why you should use imaging, or you could use imaging in oncology research. So the value for preclinical imaging really breaks down, in our opinion, in 6 broad levers. Preclinical imaging really gives new insights that you could not do with individual techniques, such as tissue sampling or blood sampling, or terminating deameling or having a look surgically. First of all, it's a comprehensive and very fast way to visualize entire pathologies. The second one is that it is a translational technique. Basically, you can fully quantify all the data coming from the image, and then it translates into data, or it can be translated to data of clinical. Let me put it that another way. It really translates into the clinic. So you can use it in IND enabling applications in your studies, etc. Thirdly, the preclinical imaging studies the interactions between physiological and biochemical processes. And then this can be done longitudinally over time. You can monitor a set of animals or a single animal at multiple time points, similarly to with optical imaging, with the added benefit that each animal serves as its own control, which adds extra statistical relevance. A fifth value is the fact that you can have an entire look at the biodistribution of new compounds. So from the administration, the metabolism, the excretion of new compounds can be fully visualized and quantified. And then lastly, which is becoming more and more importantly in this world as well, is the 3R compliance. So it's a reduction on your use of animals. If you can just have a single animal have multiple image time points instead of having to terminate your animal every study. What I would like to do is for each of these values, give a more hands-on example. These are all studies that have been performed on some of our systems. So what we can see here in this study, there was a specific line of breast cancer cells that was inoculated in a rat, and in literature it was said that this would metastasize to the brain of the animal. However, after just a few weeks, the researchers noticed that the hind legs of the animal, and then afterwards the complete animal, would become paralyzed. And without actually knowing what was going on, they resolved to using a quick 20-minute PET-CT study. So this is with a regular FDG tracer, and basically saw immediately that there was metastases throughout the body of the animal. This can be seen in two ways. So in a PET-CT study, when you do the CT image and then the PET image, on the CT you would have, for example, the clear holes in the bone here, where you can see the necrotic tissue. And then this nicely co-registers or is aligned with the hotspots of the FDG being taken up by this cancerous tissue. And if we then have a look at the fly-through of the data, where we're scrolling through the different slices, if I can find my mouse, there we go. You can see the different hotspots lighting up throughout the spine of the animal. And on two levels, you can really quantify this data. Not only on the CT, where you can measure the actual tumor volume, it's also on the values in these hotspots of the cancerous tissue that you can track over time, after 5 days, after 10 days, after X amount of months. How have these values changed over time? So besides of obtaining this visual confirmation, the value of preclinical imaging is also really in the translational capabilities or the quantification of these results. So in this study example, there were prostate cancer mice models that were used, and they all had different cell lines with a different level of PSMA expression. It is known that the PSMA expression is 100 to 1,000 times fold higher in prostate cancer tissue than it is in healthy prostate tissue or in salivary glands or liver tissue. So what we know is that this PSMA or this overexpression is correlated with tumor grade, the pathological stage, and the metastatic castration resistant prostate cancer. The aim of this study was to validate PSMA as a target for expression-based imaging. So what they did is having these different cell lines here from left to right with each a different expression level. So the PC3 at the lowest level, the 22RB1 in the middle, and the one on the right-hand side with the highest level. And really see how this specific tracer, which was a, sorry for that, which was a specific, here we are, again, an 18-fluoride isotope bound with some specific altered PSMA, how this would be taken up by the different cancerous tissues. And basically what they saw is that there's indeed in the quantification of these values a significant difference between the SUVs measured and also the tumor to liver ratios and tumor to blood ratios. So this basically enabled researchers to detect differences in PSMA expression, which is positively correlated with the tumor grade. So moving on is to highlight the effect of interactions between physiological biochemical processes, is a study where peritoneal metastases originating from the ovarian and colorectal cancer were inspected. Basically, researchers noticed with a preclinical model that the intraperitoneal chemotherapy was not as effective as they had hoped to be, because there was limited penetration. Basically, there was poor tissue penetration. So one thing that they wanted to do is to develop metastases models that could closely mimic the entire tumor microenvironment, including bulky residual tissue. And the way they did this, or the reason why they wanted to do that, is to have a suitable microenvironment to assess the tumor response on the chemotherapy that they wanted to investigate. And the way they did it is by implanting a 3D printed PLA scaffold, this device here, inside the animal, and by seeding specific tumor microenvironments and tumor cells, they could very accurately control the entire microenvironment of their animal model. And then afterwards, what they then could do is over time basically have a look at the effects of this environment on the angiogenesis, the little blood vessels that were formed. This is here seen on the highlights of the small blood vessels with contrast imaging. In a next example, where I want to highlight the fact that you can do, of course, non-invasive and longitudinal imaging, is an example of a study where researchers wanted to predict the treatment response to the combination of two different therapies for breast cancer on an animal model. So here, the uptake of, again, a 18-fluoride tracer with an FLT bound to it was measured over time. And again, you can see over time after the pre-treatment here is on the top. Then you would have the post-treatment day 3 and post-treatment day 14. The hotspots disappearing over time, and these values again can be quantified to confirm the response here. Second to last is the fact that imaging can be used to validate the biodistribution of novel compounds. So in this study, there were radio-labeled enzyme nanomotors that were injected in the animal. And they wanted to evaluate the biodistribution in vivo. So here, an animal was scanned for 60 minutes, and these 60 minutes were divided into different frames to see the tracer distribution at really specific time points. You can have a snapshot on, for example, 1 second in, snapshot on 3 minutes, 20 minutes, 40 minutes, etc., and visualize and quantify the entire process over this time. And that way, by then afterwards, really selecting in post-processing where you want to quantify the data in specific organs, you can have this result on, for example, charted here. So the specific quantification in the liver and lungs, thyroid, stomach, etc. Lastly, but this is more of an obvious point. So it is a non-invasive method. So whereas before you would have lots of termination of your animal models, for example, at the disease onset and looking inside the animal to validate everything after disease progression and after therapy monitoring. With imaging, obviously you can do this by non-invasively imaging the animal over time. So there's a reduction basically in overall resources used and on reduction on the termination of animals. Pulling that all together on a very high level, this is what for example, a study could look like. So in the planning of the study, you'd have your animal model where you select what exactly you want to investigate. You can perform the imaging for your therapy planning here on a glioblastoma model imaged here. Perform the specific therapy, in this case, small animal radiotherapy, for example. And then with longitudinal imaging, look at the effects over time and quantify the effects over time. So these were several examples. I know this is not my forte, the examples. I'm more than happy to take plenty of questions on those and provide extra material, extra information. In the back ends, we have plenty of studies and information that we can openly share with everyone. Specifically now then on how Molecubes fits in this picture. So Molecubes is a company founded 6 years ago from Belgium, and is still, in terms of the preclinical PET-CT market, still a relatively new player in the field. But we have taken an entirely different approach on how these imaging devices were designed. Basically, it resolved from the vision and mission from our founders. So our founders were all professors and PhDs from the University of Ghent. They had all hands-on experience working with these older types of incumbent instruments, and noticed how there is incredible value in imaging, but how difficult it was to work with older type of equipment. So their vision was really that imaging in preclinical setting really improves the speed and quality of research, but that they wanted to expand the use of imaging in preclinical research. It was really limited to a really specific niche of people who had the right backgrounds in physics, in radiochemistry to get things going. How they did that is by really bringing together latest technology advances, both on the hardware level and on software level. So whereas in older vendors or older models, thinking of the GEs and the Siemens of this world, who all were incredibly, definitely revolutionized this world 20, 25 years ago by introducing preclinical imaging, they still tried to use clinical-like systems, using clinical-like designs and clinical-like hardware to make these types of devices. Whereas Molecubes really designed them from the ground up and used the most recent detector design, hardware designs, and also putting themselves in the shoes of the researcher. So how can we make the working with these systems much more easier? So focusing on the user friendliness. That's basically how they came up with as we call them, the Molecubes. It's an entire platform consisting of 3 different imaging modalities. So there's 3 cubes, the X cube, gamma cube, and beta cube, which are respectively for CT imaging, SPECT imaging, and PET imaging. 2 things that immediately stand out is, first of all, the fact that the design choice was made to split these modalities. There's a multi-modular approach. What this means is basically added flexibility, both on a study level, but also on a lab planning level. On a study level, having separate modalities means that you can have CT scans going on at the same time as having PET scans going on. But if you would like to have a PET CT study, you can first do a CT study and then move the animal to a PET scanner and still register the images to have your full PET CT view. This is a big benefit in terms of throughput, and is the reason why we have some pharma customers only swearing by the Molecubes to achieve up to hundreds of static scans per day. The second reason in why this adds flexibility on a planning level is that you can easily start out with one of the modalities or a couple of modalities and then grow your lab over time as the needs and the requirements for capacity grow as well. So the second major point that stands out is the fact that they are a true benchtop model. So this is about 20 inches in each dimension, a literal cube, fits on a standard lab bench, doesn't require significant or any really infrastructural changes in a lab. Basically requires a standard outlet and an AC room, and you're ready to go. So the use, it's even though the cubes are nice in the outside, it's also really making use of the latest detector technology and the latest types of algorithms that all have been designed in-house. That means that there's no compromise on any quality of the images while scaling these systems down. Some more highlights to share here is, I think the most interesting ones are this biodistribution PET study on a full body mouse. So just in a single bed position, you can have the full field of view on the entire mouse and perform a dynamic scan on that animal. The image next to that is the use of our so-called hotel bed, which supports up to 4 mice simultaneously. Again, an incredible factor in achieving higher throughput imaging. And then the image on the right-hand side is a zebrafish, which I believe is just about an inch tall. And just to highlight again the resolution of the system, you can still distinguish the different organs within this small animal. So Molecubes, even though we're still young-ish in this industry terms, with now 6 years on the counter, we've grown quite nicely over these 6 years. We're now a team of 24 people, most of us still based in Belgium, a few people in US and in China. But now already 100 cubes. It's an outdated figure, but we have 100 cubes installed worldwide in pretty much all major countries. In the US specifically, it's really in the past 3 to 4 years that we've grown quite a lot. So this is thanks to our partnership with LabLogic. This is a company that we collaborate with so that they can provide nationwide service and support, provide the installation services of this equipment. We have a local office actually here, or at least LabLogic has a local office in Tampa, so any stock parts, spare parts can be shipped from there at any point in time. Next to large academic sites, I believe UChicago and Pennsylvania were our first commercial customers back in 2017. We are now seeing lots of uptake in even smaller biotechnology companies like Curie Therapeutics, like Early over on the West Coast. These are teams of maybe 10, 15 people but who want to make the jump into having imaging on site as opposed to working with external partners. Gives you much more control over what you're trying to achieve. We talked about the image quality and the quantification aspect about it. We've talked about the scalability, how you can add extra cubes. So one thing maybe not mentioned explicitly in this presentation is really the fact that it's focused on user friendliness. We really try to make these systems that people, regardless of their backgrounds, could work with them. So it's a simple interface, a few clicks to get scans and reconstructions going. Can achieve very high throughput scans. So for your more advanced images where you would like to do dynamic scanning, that's really for example, the biodistribution studies that I showed earlier on. You can do up to 40, 50 a day with just a PET-CT setup. Next to that, we have a great leading application team and service team dedicated to helping teams or helping clients, whether they are academic or small biotech companies that have no experience at all with imaging, to help them up and running from the get-go. Yeah, it's a small example here, one of the latest installs in Essen, just to bring home the point that these benchtop formats is super small and can fit in what looks like almost a broom closet over there in Essen. And then lastly, just to show what a typical PET-CT scan looks like, a short video here. So we start off with the there we are. The animal in an animal bed on a prepping station where you can anesthetize, prepare any catheters if there's a need to, before slotting it in the first modality of choice, in this case a CT cube. Select the field of view and the type of protocol that you would like to use on the user interface, and then start the actual scanning procedure. So for the CT, which is a fully self-shielded cube, there's LED shielding all around, and you'll see a little shutter door closing in a minute. Scanning takes between 20 seconds and 2 minutes, depending on the different protocols that there are. Get some visual feedbacks on progress of the scan. And then to have your hybrid imaging, so your PET-CT imaging, what you can then simply do is transfer the animal bed to the second modality, in this case the PET cube, which happens here. Again, via the same user interface, select the different modality, enter the parameters, and get going on the scan. So here, PET scan typically takes, depending on the study, 10 minutes up to half an hour or an hour for longer dynamic types of studies. The images are reconstructed inside our cubes themselves, and the output is a medical DICOM file, which can then be opened in any standard post-processing software out there and manipulated for further use. Yeah, I mentioned the fact that we have this application support and the service support on US ground, but also on Belgian ground to help teams out with their imaging. I think lastly, some specs, if it would be of interest to folks that I can highlight here. So the things that you want to look out for in PET and CT specifically, look here, is definitely the, first of all, the field of view in your PET studies. So the 13 centimeter actual field of view is the length of your field of view. There's an entire adult mouse can fit in that field of view, so you can have the full biodistribution or the full view in your dynamic studies. In terms of resolution and sensitivity, resolution is about 700 microns for the PET cube at a 12.6% sensitivity, which is one of the highest in the market. Now, regarding the CT cube, there the highlights are the fact that it's a 50 micron resolution in a special helical or gantry design inside the system, which makes it able to have a continuous scanning motion. And that's why we can do these types of scans in a matter of 20 seconds. It's on that note that I'd like to close, but definitely want to open the field for, or open the ground for questions. I hope I was able to share at least some highlights. Again, if there's questions that I cannot answer, I'll be more than happy to take them home and give a more other answer afterwards. But thanks for inviting me, and thanks for your attention.